Chapter 17, The Evolution of Animals. So what is an animal is the first question we should ask. An animal can be defined through a variety of criteria. One is that all animals are They are all multicellular. They are all heterotrophic. Heterotrophic. Well, hetero means different. Troph means feeding. So all animals get their food by feeding off of something else. They get their carbon from other sources, whether it's another living organism or a dead organism or what. They do not produce their own food. They're heterotrophs. They lack cell walls. No animal has a cell wall. Even the ones with thick, hard shells, that's not a cell wall. That's actually, depending on the animal, a variety of substances. And most animals have muscle cells for movement and nerve cells to control those muscles. Most animals, not all. So let's take a look at the animal life cycle. The basic animal life cycle is that male and females produce gametes through meiosis. And you get haploid gametes. And then an egg and a sperm fuse, producing the first cell of the new organism. And that first diploid cell of the new organism is the zygote. And mitosis goes from there to produce a multicellular adult. And at that point, that adult undergoes meiosis to produce gametes. So that's the basic life cycle. On and on and on. So if we're going to talk about animals, it's important to talk about the Cambrian explosion with regards to the evolution of animals. The Cambrian explosion occurred long time ago in Earth's history. And this is when animals underwent massive amounts of adaptive radiation, meaning that animals found new unoccupied niches. Remember, a niche is a role in the environment. They found unoccupied niches in the environment and exploited them. And they adapted to these new niches that they found. The Cambrian explosion showed a massive increase in the diversity of animals. And it was likely triggered by ecological interactions and a sudden increase in atmospheric oxygen. See, before the Cambrian explosion, the atmosphere was predominantly carbon dioxide dominated. But cyanobacteria came along and they underwent photosynthesis. And remember, one of the byproducts of photosynthesis is oxygen. So these cyanobacteria showed up and oxygenated the atmosphere. They increased atmospheric oxygen, and this caused, well, massive extinctions, and at the same time, it caused massive adaptation because you had drastic and large blanket extinctions occurring. So organisms that had occupied niches died off when the oxygen increased. And then these niches were open, and the animals entered these niches. And this is when the first body types were seen. Many that in fact no longer exist, including body types that have odd numbers of eyes and odd numbers of appendages on bilateral bodies. Bilateral means you can divide an organism into two parts and they will look equal. So humans are a bilateral organism. If you take a knife and just go cut me down the center there, my left and my right side will look pretty darn similar. They'll mirror each other. Symmetry, similarity. So bilateral symmetry. Two sides. So in the Cambrian explosion, we saw bilateral organisms that had odd numbers of eyes, 3, 5, 7 eyes, and odd numbers of appendages. Some of them have weird little claw appendages just coming off the head, just a single claw appendage coming off the head. looked really weird. So, what happened to all the weird ones? Well, the Cambrian extinction event followed the Cambrian explosion. The Cambrian extinction event ended the Cambrian era, and it killed 75 to 95% of all species. Of all species on the planet. So the Cambrian explosion was a sudden massive increase in the number of different species on the planet. And you had this amazing prolific period in which animal species appeared and appeared and appeared and adapted to new and interesting niches. And then the Cambrian extinction event occurred, and 75 to 95% of all of those died off. And you were left with a lot of the boring body types that you see. For instance, bilateral with one head and 4 limbs. Woo! And then, of course, there's radial, like your jellyfish. So here are some of the weird things that you saw in the Cambrian era. And there's that guy with the weird claw coming off right there. Funky. And you can see 5 eyes right there. That's weird as well. So odd numbers of eyes, odd numbers of appendages. Here's a freaking worm with weird leg-like appendages crawling along. So lots of very weird organisms that died off. And now the only evidence we have of their existence is their fossils. So, what was left, however, found a new world that they could evolve to colonize. And they filled the new empty niches. 75 to 95% of all species on the planet died. So the 10 to 25% of species left filled all these new open niches. And you had the dominance of the standard animal body types we know and love today. So let's classify animals as we know them today. There are some basic criteria we can use in distinguishing different types of animals from one another. Phylogeny is a great way. Phylogeny is a great way to organize those features. Basically, a phylogeny is an evolutionary tree developed through physical and genetic similarities and differences, basically showing how related different animals are, trying to determine maybe what animals, what lineages evolved from common ancestry, how closely related different animal lineages are, and we determine how closely related they are based on physical and genetic similarities and differences. How closely related they are versus how vastly differently related they are. So you can see that, amongst the primates, humans and chimps, very closely related. Humans and mice, not so closely related. So that sort of thing. You also develop a phylogeny based on the body plan of an organism. And this is where we get back to the symmetry. Remember bilateral symmetry and my brief shout out to radial symmetry. In addition, we have things like the presence or absence of certain kinds of tissues, the presence or absence of a body cavity, and other things. And then finally, pure genetic relationships, which are the similarities in mitochondrial DNA. Remember, the mitochondria, that organelle that produces ATP in eukaryotes, has its own DNA. Remembering endosymbiont theory? I know you are. That's right. Endosymbiont theory said that the mitochondria was once a free-living cell that was absorbed by some ancestral proto-eukaryote, and they developed what's commensal relationship or a mutual relationship, symbiotic, thank you brain, symbiotic relationship that was a mutual relationship in which the little absorbed free-living prokaryote became the mitochondrion, produced ATP, and then the proto-eukaryote that became the eukaryotic cell we now know provided shelter and energy. Anyway, because the mitochondria have their own DNA, we can see how closely related mitochondrial DNA is. See, mitochondria are sheltered from a lot of the evolutionary pressures that cause changes in the genomes of animals. And so because these mitochondria are sheltered from that, their cell buffers them from the external pressures, you see very little changes in mitochondrial DNA over time. Really, the only changes you see are rare mutations. And so, by looking at how closely related mitochondrial DNA is, we can get an idea of how far apart they are on their phylogenetic tree. So, because it doesn't mutate as much as nuclear DNA, and it's not as susceptible to evolutionary pressures. So it doesn't change that much. So if there are a lot of differences in mitochondrial DNA between organism A and organism B, that means that they had to have been separated by a vast amount of time. They're not very related at all because there's been time enough in their separate lineages for a lot of differences in mitochondrial DNA to accrue. So there are 9 major animal phyla. So remember a phylum. So when we talk about animals, we're going to have to kick back to lecture 1 where we talked about classification, which went kingdom, phylum, class, order, family, genus, species. Kingdom is Animalia, so animals. So phylum. So that is the next basic division. So we're going to be dividing all of the animals into 9 major groups. So phyla is the plural of phylum. So the first group we'll be looking at is Porifera, the sponges. Then comes Cnidaria, the jellyfish and sea anemones. Then Mollusca, which is snails, slugs, and surprisingly, squid and octopus. Platyhelminthes, which are flatworms. Annelida, which are worms you might be more familiar with. They're segmented worms like earthworms and leeches. Then we have Nematoda, which are roundworms. You might know of these by name only. Heartworms that you give your dogs pills for are a kind of roundworm. They're a nematode parasite. Arthropoda, everybody knows arthropoda, even if they don't know what an arthropod is. They've seen them because it includes insects, it includes crustaceans like lobsters and crabs, it includes arachnids like spiders, and many more. Then there's Echinodermata, which is a pretty cool name right there. I mean, just fun to say. Come on, say it out loud. Nobody else is looking. Come on. Echinodermata. 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 It's fun. Here, I'll even get an annoying song stuck in your head. Echinodermata. What a wonderful phylum. Echinodermata. Ain't no passing phase. It means spiny skin for the rest of your days. Okay, never mind. I'm getting old. So this is starfish and urchins. And other organisms of that related. And then finally comes chordata. Chordata refers to organisms with a dorsal nerve cord. What does that mean to you? Spinal cord. So a spinal cord running through them. And this includes everything else that you're familiar with. So this includes anything with bones, automatically a chordate. If it's got bones, it's chordata. That's an easy thing to remember right there. Other than that, if it's got a spine, it's chordata. So here is a phylogenetic tree of the animals with a few basic differences in here. First off, remember how I told you about the presence or absence of tissues being a way to tell different organisms apart? Well, here's where we separate sponges from everything else. Sponges don't really have true tissues. Then we can separate Cnidaria, the jellyfish, and anemones from everything else by pointing out that the jellyfish and anemones are the only organisms that have radial symmetry, the only animals that have radial symmetry. And then from there, you have all the organisms that are bilateral, meaning that you can divide them into two halves. And from there, you separate them into protostome and deuterostome, which I don't recall if I actually mentioned, but I'll give a shout out to it here in a little bit. Anyway, and then from there, you can go on to all the rest of these major groups. So let's take a look at features we use in the differentiation of animal groups. Tissues versus no tissues. Tissues are cells grouped together to perform a specific function. What does that mean? Well, think of it this way. Here's a group of cells in a layer performing a specific protective function. They are epidermal tissue. My skin is a type of tissue. Its function is mainly protective. So there are lots of different types of tissues in the body. You have the tissues lining the stomach. Some tissues produce acid to make your stomach acid. Some tissues make the stomach lining to protect the cells of the muscle cells of the stomach from the acid. So other tissues produce enzymes. So you have lots of tissues performing specific functions. That's what a tissue is. It does something. So one of the basic ways we differentiate groups of animals is whether or not they have tissues. Tissues show up during embryonic development. So here's when you go from zygote to blastula. The zygote, as we will recall, is the original single cell of a new organism. Once a sperm fuses with an egg, it makes a diploid cell called a zygote. And that zygote starts undergoing mitosis. Well, one of the things it does is develop into a hollow ball of cells. And that hollow ball of cells, think of it like a little basketball, is called a blastula. And once you get to the hollow ball of cells, you'll start to see different types of cells becoming different, sort of separating. Cells on the outside of the ball will eventually become epidermal tissue. We call it ectoderm. Ecto meaning outside. Then cells in the inside starts to fill in. And cells that sort of make up the middle area will become what we call mesoderm. And that'll give rise to a lot of muscles and connective tissue. And then finally, you'll have the cells at the very center, which are the endoderm. Endo meaning inside. And those will give rise to some of your blood vessels. The basics of the blood vessels are endoderm. So a lot of tissue is endodermal. You don't have to know the three different divisions, but basically from those three basic divisions, we then get every other type of tissue in the body. Ectoderm, mesoderm, endoderm. Actually, you know what? I think I do require you to remember that. So strike what I just said there. I'm a jerk. Okay, so ectoderm, mesoderm, endoderm. All right. Now that we've got a ball, and this ball has three different layers to it, cut your ball in half, you'll have outside the ectoderm, the middle, the mesoderm, and then the center, the endoderm. Well, next, two involutions form. Involution is one of those fancy science terms we use to sound super smart. But basically what it means is a little indentation forms. Think if you have a balloon, right? A balloon is a hollow ball. So start to poke your finger into the balloon and that's an involution. Cells at the surface start to form a little pit and move inward. So that's an involution, the formation of a pit, and then it moves inward, poking a balloon. So, and these involutions will form on opposite sides. And they'll poke towards the center, and they'll create a pit running down the center, and eventually they'll meet, and then you'll have a canal, or a little tube running through the center of the organism. And that little tube will become the digestive tract. So, here we go. This is the blastula, your hollow ball. They actually have a special name for the inside, the blastocele. Don't worry about it. But then here's that first involution. So if this is our balloon here, then this over here, this gastrula, is where we make our first poke in the balloon. We have the blastopore. So the ectoderm is tissues in contact with the external environment. Skin, and interestingly enough, neural tissue is ectodermal. So the brain tissue originally developed on the surface of this little ball of cells. And it sort of folded inward at some point. But anyway, enough about that. Skin is a great example of ectoderm. Mesoderm, connective tissue, muscles, including your heart, your bones, your urinary and your reproductive organs. And then finally, endoderm, the digestive tract, which is the tube running all the way through from your mouth to your butt, which consists of your mouth, throat, stomach, intestines, rectum, anus, and all the organs associated with the digestive tract, like your pancreas and your spleen. Your lungs are also endoderm. Your liver is endoderm. So, good examples. All right. So that whole giant thing was how we use tissues to differentiate two different groups. And if we take a peek back, everything except sponges has true tissues. Then we have symmetry. No symmetry is an option here, which is where the organism cannot be divided into identically shaped parts. So there's no way you can divide the organism and get parts that look like the others. Then we have radial symmetry, sea anemones and jellyfish. These are organisms that can be divided into identical shapes around the center. Think of how spokes divide a wheel into even little triangles. Or better yet, since you're all in college, think of it as slices of pizza. All the slices of the pizza are the same, and they are in a circle around the center. So that's radial symmetry. You can make cuts around the center in a circle. Radial. Then finally, bilateral, which I already mentioned. So an organism can only be divided into two identically shaped halves, which means it has a front and a back end. So front generally being the head, back generally being the tail. All right. So now that we've talked about tissues and symmetry, it'll be time to talk about porifera in the next video.